Welcome to the Deep Dive, where we take a stack of dense sources, pull out the most shocking and useful nuggets, and make you an expert faster than you can brew a cup of coffee. Today, we're heading to a place that should be the definition of a biological void, yet it is. Well, it's teeming with life, the Chernobyl exclusion zone.
It's the ultimate paradox. Nearly 4 decades after that catastrophic 1986 explosion, the ecosystem has rebounded in some really strange ways.
Yeah, we've all seen the documentaries, right? The wolves, the wild boar, thriving because there are no people.
Exactly. But we're focusing on an organism that isn't just surviving in spite of the lingering radiation. It actually seems to be thriving because of it.
Okay, so this is what we're deep diving into today. A strange, kind of humble, velvety black fungus. Its name is Cladosporium sphaerospermum.
And get this, it was discovered clinging to the interior walls of the wrecked Unit 4 reactor.
Arguably the most dangerously radioactive location on the entire planet.
Absolutely. So our mission today is twofold. First, we're going to unpack this wild, almost sci-fi theory known as radiosynthesis, a proposed new foundation for life.
And then we pivot completely.
We pivot completely. We're going to explore how this humble fungus, discovered in the ruins of a nuclear disaster, might just be the key to shielding you or, future human colonists on missions to Mars.
Okay, let's unpack this, starting with the sheer impossibility of life existing there at all. Before we talk about fungi feasting on anything, we need to understand what ionizing radiation actually does to living tissue.
Right.
Give us the breakdown. Why is this stuff usually so deadly?
Well, basically, ionizing radiation is energy or particles powerful enough to knock electrons right off of atoms.
Okay.
So when these particles hit a cell, they don't just warm it up, they tear it apart. They shatter the bonds of crucial molecules and, critically, they shred DNA.
Which leads to mutations, cell death.
Exactly. In complex organisms like us, that's acute radiation sickness, cancer. For nearly all life as we know it, this environment is actively trying to rip your foundational biology to shreds.
So if the environment is pure biological destruction, what did scientists even expect to find when they first looked inside the sarcophagus?
They expected almost nothing, a biological desert. But the initial shock came in the late 1990s. A microbiologist, Nelly Zadonova, and her team from the Ukrainian National Academy of Sciences did a field survey inside the reactor shelter. They were just stunned. They found a whole fungal community.
What community?
They documented 37 different species, in fact.
37? In the shadow of Unit 4. That's truly incredible.
It is.
And these weren't just random, desperate survivors, right? The sources all emphasize a very specific, peculiar common trait among the dominant species.
Precisely. They tended to be black or dark-hued, loaded with the pigment melanin. And our fungus, sphaerospermum, it dominated those samples. It had some of the highest levels of radioactive contamination.
But the real kicker was about their movement.
That was the most profound observation. The fungal filaments, or hyphae, they weren't just randomly distributed. They seemed attracted to and were actually growing toward the sources of radioactive particles.
Like a plant reaching for sunlight. What's the technical term for that? For, moving toward a radiation source.
Zadonova called it radiotropism. It literally means turning toward radiation.
Huh.
And that analogy to photosynthesis is profound because just as plants respond to visible light for sustenance, these fungi appear to be responding to an invisible, deadly energy source in the exact same way.
So we have an organism that's not just shrugging off this deadly force, it's actively using radiotropism to seek it out. This brings us directly to the core hypothesis that changes our definition of life. The radiosynthesis theory.
And that's where radiopharmacologist Ekaterina Dadachova and immunologist Arturo Casavels came to the picture. This was in the mid-2000s. They took these melanized fungi, including our Chernobyl resident, and exposed them to ionizing radiation in controlled lab settings. And they confirmed it. The radiation didn't harm them.
It helps them.
The fungi actually grew demonstrably better.
They weren't just surviving, they were flourishing. Okay, let's get to the mechanism here. How does a simple black pigment suddenly become an energy harvester?
The proposed mechanism is radiosynthesis, and it really does mirror photosynthesis. In plants, you have chlorophyll, right? It absorbs light, converts that energy into chemical energy sugar to fuel metabolism.
Okay.
In these fungi, the pigment melanin seems to act as the energy transducer. It absorbs high-energy ionizing radiation, specifically gamma rays, and supposedly converts that destructive energy into usable chemical energy.
That sounds like turning a fire hydrant blast into a drink of water. It seems impossible.
It does.
Yeah.
But the evidence supporting this conversion idea is pretty compelling, especially around electron transfer. It's the closest thing to a smoking gun they've found so far. Data found that these fungi grew up to 10% faster when exposed to radioactive cesium compared to the control groups.
10% is significant.
It is. But the real metabolic evidence came from observing what happened to the melanin itself. They exposed it to high radiation levels in some experiments, 500 times the normal background level, and this rapidly altered the chemical properties of the melanin.
And what did this alteration do, metabolically speaking?
It caused a huge increase in the rate of melanin-mediated electron transfer.
Which is a key step in how an organism processes energy.
Exactly. Any organism's ability to process and utilize energy. They found this rate increased 3 to 4-fold.
Wow.
If you think of a plant's engine, that's the metabolic equivalent of suddenly generating 300% more power from the same sunlight. It suggests the radiation is fundamentally changing the energy output of the organism.
Wow. That completely changes the organism's relationship with radiation. It's not just a byproduct they tolerate.
Right.
It's a primary energy source they metabolize.
Yes, and this is why we have to draw that sharp distinction. These fungi are not just general extremophiles, which survive in spite of severe conditions.
You're different.
They are proposed to be radiotrophic, meaning they potentially grow because of the radiation, utilizing it as a viable energy source, much like we use food. It's a completely different rule book for survival.
But as with all truly revolutionary biological ideas, there's a scientific roadblock, a catch. Radiosynthesis is still, at the end of the day, only a theory. Scientists see the evidence of accelerated growth, but they're missing the complete pathway.
That is the crux of the debate. Despite the strong correlational evidence, you've got the enhanced growth, the increased electron transfer, the radiotropism, the precise biological mechanism remains unproven.
So what are the specific missing links? What do they need to find to prove it?
The two biggest hurdles involve carbon. First, they haven't been able to show carbon fixation dependent on ionizing radiation.
Okay, what does that mean?
It means that if these fungi are using radiation as an energy source, they should be able to take carbon dioxide and convert it into carbon compounds, like sugar or biomass, just like plants do with photosynthesis.
Ah, that's the gold standard of proof.
That's the gold standard. So they see the engine revving faster, that's the electron transfer, but they haven't seen the final product being generated purely from that radioactive energy.
I see.
They also haven't mapped out the full energy harvesting pathway. They need to identify the reduction of carbon compounds that would prove metabolic gain directly from the radiation source. Until that full chemical chain is documented, the scientific community is, highly cautious.
And complicating things even further is the fact that this behavior isn't universal. It's not in all dark fungi.
That's a critical point for the skeptics. If radiosynthesis were some universal principle, you'd expect every melanin-rich fungus to show the same growth enhancement.
But they don't.
They don't. A 2022 study showed no enhanced growth under radiation in another melanized species, Cladosporium, cladosporioids.
So what does that tell us?
It tells us the mechanism is either highly specialized, or maybe it's not a metabolic feast at all.
So we have two competing theories here. One, they are truly harvesting the energy. Or two, the radiation is triggering some kind of extreme defense mechanism that just looks like super growth.
That's the alternative interpretation.
Yeah.
That melanin is simply an incredibly effective shield. The fungi that are melanized survive the radiation, while the non-melanized fungi just die off.
Okay, that makes sense.
So the ones that survive can then better compete for traditional food sources, carbon in the environment. In that scenario, the enhanced growth is a secondary effect of superior survival, not a direct result of energy harvesting.
We also know that producing that dark shield, that melanin, must come at some kind of metabolic cost.
That's the third point the scientists make. In the absence of radiation, non-melanized fungi sometimes grow faster than their melanized counterparts. Melanin is a complex polymer. Building it requires energy.
So it's an investment.
It's an investment. It suggests that producing that dark shield comes at a cost, unless a constant source of radiation is present to tap into, making that investment worthwhile.
That is a fascinating nuance. So while the scientists argue over the footnotes of the biological pathway, the engineers are already looking up.
Right.
Because if this fungus can survive the worst radiation on Earth, perhaps it can survive the invisible storm of deep space.
That's the transition.
Huh?
For astronauts undertaking deep space exploration, whether it's the Moon or Mars, cosmic radiation is consistently called the greatest hazard.
Right. We're protected here by Earth's magnetic field, but once you leave low Earth orbit.
You're exposed. It's an invisible bombardment of charged protons from distant exploding stars. This radiation hazard dictates everything: mission length, shielding costs, astronaut health.
And this is where the engineers realized that maybe the solution to the biggest modern space challenge was growing on the walls of a defunct Soviet reactor.
Precisely. NASA and others are desperate for lighter, self-regenerating shielding solutions because hauling metal or thick polyethylene into space costs thousands and thousands of dollars per kilogram.
So this fungus offers a potential biological answer.
It does. The research team, led by Niels Avrish, decided to put sphaerospermum to the ultimate test and sent it to the International Space Station, the ISS, back in 2018.
What were the metrics they were looking for up there? What did they want to find out?
They monitored two main things: growth and radiation attenuation, or blocking. First, the fungus grew really well. It actually achieved an average growth rate 1.21 times faster in space compared to the control samples on Earth.
Faster in space.
Faster. Though Avrish noted that zero gravity may have contributed to that, which complicates the radiotrophic claim, but the main thing is the growth was successful.
But the real payoff was the shielding capability, even in that high radiation environment.
Absolutely. The researchers grew the fungi in a patch and placed a radiation sensor directly beneath it. They found that even a thin layer, just 1.7 millimeters thick, was able to reduce the measured radiation dose by 2.17%. Wow. For something less than 2 millimeters thick.
It's a remarkable protection capability for something so thin and lightweight that you could effectively paint onto a surface.
This sounds revolutionary for building human outposts. If we can grow our walls and protection on site, we solve the tyranny of launch mass.
That's the concept of myco-architecture. And the protective potential is staggering. They calculated that a 21-centimeter thick layer of this fungus, grown as a continuous shield, could almost negate the annual radiation dose that future colonists would face on the Martian surface.
Almost negate it? You mean bring it down to Earth-like levels?
Effectively, yes. It brings the radiation exposure down to Earth-based levels.
That is the dream, but let's get real for a moment. Growing a 21-centimeter wall takes time and resources. What are the major engineering hurdles here?
Of course. Does this fungus need Earth nutrients? I mean, if we have to ship everything to Mars anyway, what's the net gain over just hauling polyethylene?
That's the critical engineering challenge, and it's why the research is focusing on using local resources. The concept is that the fungus could be grown using Martian regolith, the soil, as the substrate.
Ah.
You might need to ship some initial nutrients, but the primary mass is sourced locally. The fungus, enriched with melanin, then rapidly grows into dense, self-repairing, high-density walls using biological processes instead of energy-intensive complex manufacturing.
So we reduce the launch mass by relying on biology to do the heavy lifting when we arrive.
Precisely. The sources highlight that mixing just 9 centimeters of Martian soil with the melanin and the fungi creates a viable substitute for much heavier, traditional shielding materials. We're talking about reducing the mission mass for shielding by orders of magnitude.
It's life, once again, finding a way to solve our most complex engineering problems through elegant efficiency.
It really is.
This journey is incredible. From clinging to the walls of the world's worst nuclear reactor to potentially building the first human settlements on Mars. And the key to all of it remains that fundamental, mysterious pigment, melanin.
Potentially converting a destructive force into sustaining energy, or at the very least, into protective power. What stands out here is how this discovery pushes the very boundaries of life. We're so focused on photosynthesis as the foundational energy structure for Earth, but the existence of radiotropism suggests another, maybe parallel pathway that we're only just beginning to understand.
Which brings us to the final provocative thought for you to consider as you wrap up this deep dive. If this radiosynthesis pathway is real, could it be far more ancient than we think?
That's the hypothesis. The radiotrophic life might have been far more widespread during the Hadean Eon, the earliest era of Earth's history.
So billions of years ago.
Billions of years ago. During that time, geological instability and phenomena like natural nuclear fission reactors may have made ionizing radiation sources far more common than they are today.
So this black fungus from Chernobyl might not be a modern anomaly.
It could be a remnant. tapping into one of the oldest and most robust survival mechanisms on Earth, just waiting for extreme conditions to be reactivated.
It makes you wonder what else is dormant within the genetics of life, ready to switch on when the environment turns hostile.